Hello students, very good evening. Welcome to the Proteomics lectures for today. Sorry, yesterday was not fine, so couldn't able to join. But today, let's start a fresh start. So today till 8:20 you're allowed to join. Afterwards, I will not allow anyone to join in. So whosoever have joined it till that time, it's okay, because otherwise, streaming will start. Presentation will be started then. So before we start, I just want to know from where you are. You all students, from which university, and um, in which year you are, whatsoever you're studying in. So I will be happy because I see a lot of students are not satisfied these lectures because of the complicated things in it. So I would be happy to know your uh, from which year you are and what are you studying. Could you please write in the chat box, please? So I will be happy to take your questions on the while chatting. It's create a big chaos if we start opening, taking each questions from um, while making you unmute and you're asking me the questions via speaking. So it's all better to go with this chatting type. So second year microbiology, first year University of Hyderabad. Okay, okay, MTech. Bangalore University, second year BTech. MSc, okay. So mostly it seems from wide variety. Third year, okay. Bhagakot, second PhD. Wow. First year MS in bioinformatics, second year IIT PHU. I share my screen with you also. You can see. I'm at university. Botany honors. Third year cellular molecular biology MSc. Great, great. Pursuing BSc honors third year. So you are mostly somewhere MSc and BSc students. Some of you could see some PhDs from the IIT BHU. Third year in biotechnology, BTech students, BSc students, MTech students, BSc students, MSc students. Uh, okay. So the topics that I had shared with you today for the internship point of view. So these are the topics of your session of your this proteomics. So there are a lot of topics that I have choose so that you can choose whatever you feel like comfortable. Um, just go through them once. So it's include techniques also, and everything is there. So just go with it. So just create some groups together, and then go for that then. Okay. So some questions are there. Why 1D ST gel? The electron the complexity of protein. 2D is. I mean, 2D is dependent upon. So we will clear your question on this today for sure. We will clear this question in today's lecture. So just wait. So I have presentations for the same today. We will be going through that. So students, please join in. So I will put till 2010. So I have already started from 7:45 and it's 2010 now. So by 2010 we will start our session. So just let you all add in. So here your questions will be discussed today in our upcoming lectures. So don't worry. Sure, sure. So I will finish one session, then I will come back to your chats and we'll look for it. Uh, this way we can go for like that. So in 10 minutes, whosoever joined, they will be part of the session. Otherwise, later it's not possible. So just tell to your friends uh, because I have to come back and forth to join in. That's what I irritated part of Google Meet. Um, so we'll just wait 10 more minutes, then we start the session. Naveed Anjum, you don't have to leave the meeting. You can keep this on at same time as you do. Yeah, possible. I was told by our seniors that we have to keep it via Google Meet, so I prefer YouTube always. So can't you join? Why? What's the problem? So I will share you the link again. I will share the link again. Yeah, YouTube sessions are the best. I also not in favor of you Google Meet. So everyone joined. It's 8:10. I think we can start now. So far, from the previous days, we have done with the introduction of proteomics. So students, one thing I have to clear with you. So my style of working would be that I will be teaching you the objectives that you have signed for, objectives of proteomics. So more students wants to join in. So it's 2011, so I will not let anyone add more people. So this was much. So whatever these topics that have been discussed at the initial part, so we will be teaching you. So they are of um, what do you say, of very high end topics. So please have some reading before we start, because it will be difficult for you to understand them. So we are done with the basics of protein crystallography and X-ray diffraction. We are done with the introduction to proteomics. So I will be dealing with the 2D gel and isoelectrofocusing, SDS page, and all these techniques in details today also. And we will also deal with the chromatography techniques, which is not written there, and protein centrifugation, protein purification, and yeah, so that topics will be done. So, so I have a feeling that uh, you cannot um, understand the tough parts. So whenever I go to the higher end, things started to get really complicated for you. So please go through some books for the same. Go through some readings regarding the same, because what I have created this topic of proteomics is mainly focusing higher end also and the basics end also. It's a combination of both. So to said so, let's start with other part now. Protein stability is done. Introduction to protein is done. Protein structure crystallography is done. Now we have to start with protein stability. So on that day we are done till here, till 42 slides we were done with the renaturations. I guess, yes, on the first day. Okay, as soon experiments were done. So to continue with the next part, functions of protein, storage and transport. So what are the storage proteins? Wait a second. So plants accumulate storage substances such as lipids and proteins in certain phases of development. Storage proteins in both vegetative and reproductive tissues serve as a reservoir to be used in later stages of the plant development. So these are the various storage proteins such as lipids and proteins in various phases of development. They are available, and they are both vegetative and reproductive tissues, which serve as reservoir for the later part of your work for the storage. So accumulation of storage protein is thus beneficial for the survival of the plants. Storage proteins are also important source of dietary plant proteins. Storage proteins in wheat, the two majors are such as in endosperm, gliadins and glutens. So these are the two examples of it, gliadins and glutens. And ferritin is also a storage protein which stores iron as a ferrin. Fer means for the iron. Uh, sorry, iron, which releases its control fashion. And further, body has a buffer against iron deficiency. Um and ferritin can help to store the excess iron. So ferritin helps to, if whenever there is a loss of iron in our body, so ferritin is the one which could help us to store it back again. So this is the iron stored in the inside the ferritin. So this is your molecule of ferritin, and inside is your iron is present like this. So large molecules are those with the charge needed to help for the protein to the cross cell membrane, and proteins from a channel molecules move through the doorway from higher to low concentration. In the facilitated diffusion, they move from higher to the lower pathway. So each channel is specific to particular type of molecule in that case. An active transport is the one which helps on or relies on the transport proteins and is able to transport substances against concentration gradient. So they are the one in which the traveling is done against the concentration gradient. And this is because your cellular energy, ATP, that is present in the proteins, that can play an integral role in the function of the cell. That's why they are moving against the concentration gradient. Many are so many are embedded. Um, so many are embedded in the cell membranes or span entire the life's lipid bilayer, where they can play an important role in the recognition of signaling and transport. So hemoglobin is a protein that makes blood red. It composed of four protein chains, two alpha chains and two beta chains. Mainly they have four protein chains. One is alpha chains and one is two beta chains, each with ring-like heme group containing an iron atom. So oxygen binds reversibly to these iron atoms and is transported through the blood. So this is how your four alpha chains are made, and this is how your heme groups looks like, the oxyhemoglobin. So hemoglobin structure, they are responsible for much of the gaseous exchange which occurs through human respiratory system. So that was something little bit about some groups that are helping us in storing the proteins and facilitated the fusion, activated the fusions, and storing protein in the wheat, storing protein as a heme group, and also for the transport. So we just discussed that part in it. So protein functions in defense system and cell signaling. So how protein is working as a defense system, also as a cell signaling department. So for defensive proteins are better known as antibodies. So defensive proteins are your antibodies. And these are the key part of our immune systems, and they are formed by the WBCs and fight off the infections and the viruses. Defense proteins such as antibodies, which are known as immunoglobulins, and one of the class of proteins present in serum and cell of immune system which functions as antibodies and immunoglobulins. So antibodies set off a reaction inside the human body that works to fight off the offending organisms. So often people believe antibodies actually destroy bacteria, but it's not like this. But the direct direction is usually more along the lines of the rendering the invading organism harmless. So example of defensive protein is salivary defensive proteins actually. So it's not just like antibodies are attacking the bacteria, it's just that the system is another way around. So for example, we're gonna discuss it here is with the salivary defense proteins. So saliva is a body fluid secreted by three major of our salivary glands: parotid, submandibular, and sublingual. So these three different salivary glands that we use to secrete our saliva in our mouth. And parotid is the major salivary gland in humans, and the two parotid glands are present. Uh, and the word parotid means it behind the ear. So they are behind the ear as they're present. That's why their name is parotid gland. Then submandibular, that are located in the floor of the mouth. So they weigh around 15 gram and contribute around 60 to 70% of unstimulated saliva secretion. Then sublingual, they are the smallest and most diffuse, and they provide only 3 to 5% of total salivary volume. So there are numerous defense proteins present in the saliva. I mean, they say that in the morning, whenever you wake up, without doing your brush, you should drink a glass of water, because all the saliva that in our mouth is, it goes back into our body and it's act like as a nutrient source of nutrients to us. So that's a saying actually from our old times. And if even if you see the dogs, whenever they had, what do you say, they have any infection on their legs, or if they have any problems on their body, they start to lick it with it. So it's actually, our saliva has a lot of defensive properties, so we can heal ourselves. Even though we, if in a previous time, whenever somebody got hair wound on the skin, they used to treat it with their own saliva. So saliva has a lot of healthy defensive system in our body. So some of the defensive proteins like salivary immunoglobulins, salivary chaperokines like HSP70, HSPA, heat shock proteins, are involved in both innate and acquired immune activations. So salivary chaperokines, salivary enzymes that are a major role in the defensive system. So salivary cationic peptides and other salivary proteins like lysozymes, PPI like PLNUC proteins, salivary amylase, protein-rich proteins, mucins, peroxidases, lathrins. So they are the major examples of our defensive proteins which are act as a cationic peptides, and they are the mainly responsible for our innate immunity, and they are present naturally in our saliva. So there are five primary defense networks of salivary proteins in whole saliva. The first network may be responsible for microbial agglutinations, and this network may include these saliva proteins and peptides which bind the bacteria. In the second case, the second network responsible for lysis of microbial membranes, and these network primary targets bacteria and are likely to include salivary and cationic peptides and lysozymes. The third and the fourth network responsible for the antifungal and antiviral properties of saliva respectively. So these networks may include several salivary proteins exerting various antifungal and antiviral properties respectively. And the fifth and the last, they're responsible for immune regulatory. This network is likely to be included in all those salivary proteins which exerts immune activator and modulator properties. This network may be important for the fine regulation of the local actions of the mucosal immune system. So cell signaling, cell typically communicate using chemical signals. These chemical signals are proteins which produced by the sending cells, often secreted from the cells released into the extracellular space. There they can float like messages in the bottle over to the neighboring cells. Not all cells can hear a particular chemical message. In order to detect a signal, a neighbor cell must have the right receptor to that signal. Whenever a signaling molecule binds to the receptor, it alters the shape or activity of the receptor. So they are triggering a change inside of the cell. Signaling molecules are often called ligands. So we are done with our defensive part and cell as a signaling part in this part, in this protein function, how it can act as a defensive system. and how it could be acting as a cell signaling pathway. So to done with that, let's come to the next part, that is protein purification. So this I have a full lectures for that. We will be dealing with from how you can have a protein from the native states, from the biological sample, or from a recombinant protein. So methods of, so this is just a small touch to it, so we will not go in much details. So I have a lot of virtual labs for you today where we will be interacting with the various protein purification techniques and how you can do it. Basically from chromatography to the 2D gels, 1G gels, STS page, electrophoresis, all that things we'll be dealing with today. So in this case, and this is a small summary of that, the extraction, first the protein is brought into the solutions and by freezing and thawing. Sonication is something with the sound where you extract your protein. Homogenizations by high pressure or permeabilization by organic solvents. Then soluble proteins will be in the solvent. After that, protein is separated by centrifugation. So in bulk protein purification, the first protein to be purified are water-soluble proteins. Sodium dodecyl SDS can be used to dissolve these cell membranes. So how you do it? The precipitation, you isolate the proteins, is precipitated with the ammonium sulfate NH4 2SO4, then increasing the amount of ammonium sulfate and collect the different fractions of precipitate proteins. So fractional precipitation of proteins is shown to you here. So protein of interest is red and proteins with similar solubility are mixed, the blue ones. So precipitate contaminants, they come downwards and discard the pellets, and then you have pure red proteins with similar solubility into that. This could go for chromatography, or this is also possible that you precipitate the proteins of interest, you precipitate your proteins of interest down and discard the supernatant and resuspend the proteins, so like this also. So both are possible, and from here you can add precipitant, centrifugation, discard the supernatant and resuspend the proteins. To have a like ultra centrifugation, to do a higher level of centrifugation, so you use ultra centrifugations to have a angular momentum which could yield outward force to each particle that is proportional to its mass. So suspensions of particles are spun in a centrifuge as a pallet may form at the bottom of the vessel, and then the remaining particles still remaining most in the liquids are called supernatant. The supernatant is removed from the vessel. Then the fourth and the last one is the chromatographic methods. The basic procedure in the chromatography is to flow the solutions containing the proteins through a column packed with various materials. So different proteins can thus be separated by the times required to pass the column or the conditions required to elute the proteins from the column. Usually proteins are detected as they are coming off the column by the absorbance at 280 nanometer. There are various chromatographic techniques to purify proteins like size exclusion, ion exchange chromatography, affinity chromatography, affinity chromatography, HPLC, high pressure liquid chromatography. So I will skip this part because we will be dealing with them in details. So I will be dealing with them in my next part, protein structure determination. So this we have done already to determine the structure of protein by X-ray crystallography, and we can also do it with the NMR. So determining the 3D structure of proteins help us to understand mechanism of action of proteins, how it functions, how proteins interact with other molecules, how they perform catalysis in the case of enzymes, and interaction of proteins with other molecules including protein itself, and then miscoding or misfolding of proteins associated with the disease. So three methods used for structure determinations, that is crystallography first of all. We have done that part in details earlier, so I will skip this one also. So majorly this whole slide of 110 slides is give you the very wide variety of basics part of also proteomics, and also high end part of proteomics, or the difficult one. So it covers the basics and both difficult part all together. So we are done that. You have X-ray beam, it goes to crystal, X-ray crystallography, you have detector, then you have a computed image of atoms and crystals. So protein data collection, structure solutions, we all done with that part. Then comes the nuclear NMR, protein structure determinations. So NMR is a spectroscopy which is based on the absorption by the electromagnetic radiation by nuclei of the atom. So in this case, in NMR, your nuclei of the atom, it absorbs the EMR radiations, that is your electromagnetic radiations. So protein nuclear magnetic resonance spectroscopy is one of the most powerful tools for elucidating the number of hydrogens or proteins in the compounds. So somebody wants to get in again. So, so it is used to study wide variety of nuclei like H1, uh, 13 carbon, 15 nitrogen, 19 fluorine, 31 P. So it helps us to study variety of nucleus. So a major set of distances between atomic nuclei. So it's majorly find the distance between the various nuclei. Why? For proteins that are hard to crystallize, which are not possible by X-ray crystallography, and for proteins that can be dissolved at higher concentrations, that are also possible with NMR. And to study the dynamics of proteins like conformational equilibria, folding, intramolecular and intermolecular interactions. So these are all things that could be done with the help of NMR. So this is based on the nucleus spin, which has a angular momentum vector. Spins can be parallel or anti-parallel externally. Magnetic field and applying radio frequency change this state. Steps includes protein solutions, NMR spectroscopy, sequential resonance assignments, then conformational constraints collections, and structural calculations. So in first, you do the isotope level protein preparations. So in the section A, you have isotope level preparations, and then you collect the NMR data. Then signal assignments are done in the third section. Then simulated annealing with NMR based restraints. Then validating of your precision and accuracy of structural models. So we will also go through the NMR data later. So this is just to tell you a little bit about have introductory class today to understand about each and every aspect of proteomics, which includes in nutshell. That's the basic part of it. So protein solution, highly purified protein solution is prepared with 300 to 600 microliter with protein concentrations. Data collection is done. Distinct nucleus produce chemical shift in two main experiments. One where magnetization is transferred to the chemical bonds, and one where the transfer is through the process. Then sequential resonance assignment is done, as we discussed earlier. Then collection of conformational constraints to calculate the distance between the nuclei, angle between bonds, motions in solutions, chemical shifted. So it's telling you about more conformational data later on. Then you calculate the structural calculations, which gives you an ensemble of a structure. Then comes the cryo-electron microscopy, which is a new technology for study the architecture of cells, viruses, protein assemblies at molecular resolutions. So when a beam of electron is passed through specimens, a part of it is transmitted, and this part, when projected on the fluorescent screens, its image can be seen by the observer. So mainly the electrons are passed to the specimen, and the transmitted part is projected on the fluorescent screen, and from that what you see is image of your structures, of your barriers, or any cells or proteins. So biological specimens include thin film vitreous sections. So you have a sample, you put them on the grid, and then you freeze it, then you check by electron microscope, then you collect the data, you make a 2D projections, you pick particles, particles are aligned and averaging is done, you make a 3D map, and you have a 3D model of your protein or your structure at the end. So what are the advantages? It's allow the observation of specimens that have not been stained or fixed in any way, showing them in their native environment, less in functionally irrelevant conformational changes. Disadvantage is expensive, resolution is not high enough in that case. So that's it for this part. I think you don't have these lectures with you, so I will share with you already now. It's in the day 3 one. So student, just take a 5 minutes break. uh, so let's say it's 38, we meet at 42 or so, 43. This slide is the bigger one. So just give me, yeah, you will get these slides. I already sent it on WhatsApp. So give me 5 minutes. I see you soon then. So, students, so now before I go ahead, let me share some more paths with you, or some students sharing the PPTs. So we will start with the proteomics basics based on the mass spectrometry. So that was done during the time at the Germany when I was there. So I will be sharing some presentations from there that time. So basically, this is showing you a small to large biomolecules, what drives the development of mass spectrometry. So here you can see a very nice overlapping of six. There are like overlap proteins that have been discovered, like 6004, 667, and they are known. And in total, around 19,000 proteins has been exist so far. Just a second. Just a second, students. Yeah. So far, mhm. So less proteins. So I can say there are still less proteins have been discovered than the proteins it has to be. So this is by Geiger et al. Around 131 proteins have been discovered. Then Shiromuzi in 2013, 813. Human body map around 722. Marin et al. In the nature method have discovered 323. And Mohammad Golami et al. In the cell reports have discovered in 2013 around 703. So still proteins are quite less discovered than the genes so far. And this is showing you a very nice protein networks of Streptomyces cerevisiae. It's a interactome, which is a cluster with each other. The protein clusters with each other. It's showing all the interactions, pathways, they're coming together, and how things are coming in that case. So interactions and components and a single complex has been shown. So to measure measurement of intact mass is no solution. If you can, so there are once molecule is detected, it could have a different charge. So these are the molecular masses that you have discovered at the different ranges with a different charge. So calculation of molecular mass detection of the isotropic peak and no heavy isotope. So this is one molecule which has charge of 13, 12, 11, 10, 9, 8, 7. So here you can see the charge of 1, 2, 13, 12, 10, and each charge is giving a different mass ratio, different peaks. So that's how different charged. So in the at the end, what you calculate in your mass spectrometry is calculate mass into charge ratio, m by z. Mass is to charged ratio, that is m by z. Mass is to charge ratio. So these are all isotopic charges of a sample, and with corresponding to that you have different masses. But when you divide them, you will always have the same value. That's the beauty. If you divide them with them, you will always have the same value. So true mass is the value multiplied by charge. The true mass would be value multiplied by the charge. Multiply by charge. So this is showing you to start your to mass spectrometry, the classical in-gel digestion approach, in which you have to identify your proteins. So you separate the proteins on the 1D gel first of all, and then like in this case, this is 4 to 20% Vitreous gel, and the lane is approximately contains 20 microgram of your protein, and it is a Comesi staining is better than for MS than silver staining. And then this is further cutting pieces plus de-staining is showing here like this, and then proteolytic digestion is done in this way, and then further the gel pieces are at the bottom, and the peptides excited from the gel pieces are at the top. So your peptides will be at the top after the proteolytic digestion, and most of the time your proteolytic digestion is done with the enzyme called trypsin. And trypsin cleave the sites of description endoproteinases, and specifically, so there is an enzyme amino acids called R and K. So specifically the trypsin is cutting that, which is mostly used in our cases, in our the ionization and fragmentations of our stages. And hardly we use lysine, aspartic acids, glycine, chromotripsin, arginine. So we rarely use these kind of trypsins for our mass spectrometry experiments. So whenever if you are gonna start your mass spectrometry experiments in future, so these slides will really help you to understand what you are really working for and why you are working for it. So to start the mass spectrometry, first you have to purify your peptides and go for LCMS analysis. In earlier cases, liquid chromatography mass spectrometry analysis, it's known as, we use simple LCMS, but nowadays we are using nano LCMS MS. So two times mass spectrometry is done, and also nano type of LC is done at the moment. So these are your peptides to purify your proteins. These are stage tips which have column inside it, C18 column, and we have a filtered resin in it. And then your salt contaminants, you add your peptides, then you add desalting agents like 60% acetonitrile and 0.1% of TFA. And then at the end you have a desalted peptides, and then you go for the nano LC MSMS to get the peaks for your experiments. So there are two types of studies that has to be done at here. First for the mass spectrometry, second in silico endoproteinases. So first, in the first case, you have protein like this, and you cut it with the help of endoproteinases such as trypsin, and you have lot of small peptides of your protein. Some are bigger, some are small, some are in this shape, some are in this shape. Then you do your MALDI-TOF. In this case, they done the MALDI-TOF, and you got the various peaks. On other side, you can also, you have to do is you have a gene, and you do the in silico, like with the help of bioinformatics with the computers, you digest them, and you have many peptides also, and then you compare these two peptides for your results, and then you come out to be one conclusion at the end. And then identification of gel purifier proteins via peptide mass fingerprinting. So if one protein is most abundant, easily identification. If two protein is equal density, identification is possible. If higher complexity of a mixture, identification of any protein is possible. So protein 1, 2 and 3, we have identified protein 1 in that case, and protein 2 and 3, they have very low concentration and we couldn't find them. And similar to that advent reagent, so similar to advent sequence peptides could be ladder sequence. So ladder sequence means like this: 1, 2, 3. So there's a ladder like of sequence are happening. As the bigger the peptides, bigger the intensity, smaller the peptides, smaller the intensity. So like this, it's going on like smaller, smaller, smaller. But complex mixture of peptides cannot be purified to 100% at that. So sequence, here you can see the fragmentation of the sections, measurements of molecular weights. Uh, so here mass plus charge ratio, M2 plus signs, and measurement of the fragments of the molecular weight. And first peptide are then how to analyze this data. Once you're done with your purification of proteins, they are added into the peptides, desalting is done, then you have to go for fragmentations. And the fragmentation is done using the Beiman et al. It's a German scientist who identify from the LC separated peptides, and you have to make an MS peptide fragmentations. So this is how it is done. So you have X, Y, Z. So if it is cut from this part, from the carbon of this before the carboxyl group, then it's a X3, and the bottom will be the A1. So this way you will have B1, B2, B3, and Y1, Y2, Y3. So you have many ions arranged like this of a peptide in this way. So we mainly need Y ions and B ions at the end of your MSMS, and this is showing us now data of total ion kind, total TIC. This is a total ion kind, and this is the extracted chromatography of the same data. This is extracted chromatography, and third one is showing us the MSMS data here. So here we can see the peak is being bit large here with this one, and we could see with the total ion kind extracted chromatography data seems to be coming out here, and we see the MS data to be also coming out to be here. So this proves that our work is going fine. We are working on the right track. So at the end is the mass spectrum charge of the same. So here Z equals to 3, 546.89, the value is here. So that is somehow is the peak that we were looking for, or also the Z1 here, the 533.19. So peak loss intensity. Then afterwards we go for raw data. We check each thing with the mascot. Mascot helps in silico digestions. So like I said you that in silico digestions could be done with the help of MSMS data to do the search engines, and peak list is done with the help of this. So you put your data and it gives you all the values of which peaks belong to what mass and which peptide does it belong. Then you compare this data with your previous data here, and then you can compare both data to bring out the real analysis of it. So this is the sequence coverage that you can see finally after the results are given out by the mass spectrometry. So this is data given by Peak software. So where your peptides of one protein. So you can see here. So this is the amino acid sequence. Whole this is one is amino acid sequence, and the blue lines is the one with the gray part, that the peptides that have been identified by the mass spectrometer. The rest is not identified because it's very hard to identify whole of your amino acids, whole of your peptides into mass spectrometry at the one go. So it's very hard to have all your amino acids, peptides to be categorized and to be identified at the same time. So that's why we have very few, and the rest things that you're seeing is the post-translational modifications. So like acetylations, cyclic arrest, carbomylations, deamidations, DDT oxidations. So these are happening at the various amino acids like here. So we can see some RU. So U is URAS, U cyclic arrest. So these are the various post-translational modifications that are happening. And here you can see the red dots and the blue dots. The red dots, this is an error plot that they have created. This is the ammonium ions, B ions, B minus H2 ions, B minus NH3 ions, A ions, A minus H2O, A minus NH3, carbon, B2 plus, B2 plus, sequence, XY, XY, YH2O, YNH3, Z, Y2 plus, and like this. So you have around 8 to 128 and 128 here. So the red ones we are interested with the red values, and Blue ones we are not so much interested. So red ones, we can see they are quite near to the errors, zero errors. So that means our data is pretty good. So they have to be near to the error, this part. Always, always. And ESI MS detector. So this is the type of detector types, QT of. This is showing you the internal part of it, that it has QT of is a detector type, which in the ESI electron spray ionization MS, which have high resolution full MS scan. So in this you have hex 1 and Q1, hex 2, then hex 3, then you have a push plus detect. Then there's a three different particle cells that you're looking for. Then Q1, Q2, then ion pusher is there, then there's iron mirror, which leads to go to the top part of a detector plate. So if you have three different particles, so they will do in three different forms. First, the green one will go like this, then the red one, then the blue one. So you can analyze your three different data in this way. This is an Orbitrap mass measurements to do so. This is just to tell you that how the ESI mass spectrometry equipment looks like, and how the internal ESI source belongs to this, and rest of the C-trap ion collection and RBMS. This is for the higher advanced people who will be doing mass spectrometry in the future. The rest part in the back is dealing with the RB for MS and HCD cells. So here we can see a difference between orbit comparisons of MS between QTF and Orbitrap, how the difference looks like. So in the first case, in the first diagram here, we can see that Orbitrap has higher proteins IDs. They have higher identification in that case, whereas QTF has low. QTF has low also. But on the other case, in Orbitrap, you have higher mass accuracy, but here's the increased speed of MSMS actually. So low MSMS resolution using the linear iron trap part. So and to coverage the sequence, we have more sequence being arranged in this part and less sequence being arranged in this part. But we get Orbitrap part is the more quality work in that. So more quality work has been taken care. And QTOF, there are a lot of errors are coming up. You can see some blue dots coming to be blue below the error, and but in this case most of the things are nearby the errors, zero error. So Orbitrap is again an advanced version of that. So Orbi is faster for low resolution MS data. OrbiXL is more sensitive in combinations with ultra high resolution MS plus low resolution MS data acquisitions. QTOF is faster for high resolution for MS data, and QTOF is more sensitive in combination with high resolution of MS plus medium resolutions with MS data. So that's it for this part. I will share with you this. Any questions, students, on that? So, Bache, we will deal with this, Ankita, with SDS page. Um, we can purify initially in this case, um in the case of mass spectrometry, we mainly done with the stage pips with C18 by desalting them. And beyond that, you can purify your proteins by many other methods, by chromatography, cell exclusion methods. So HPLC, nano LCMS, liquid chromatography. So there are many other methods to do so. Okay, uh, let me take. So we are done with this topic. So let's go to the another topic now. So I maybe yeah, Western blotting is done to see your bands actually. So yeah, tell me. So how can I just, yeah, but which slide? What is your question actually? So um, I mean there are a lot of slides. Which part shall I make like this to make it better? So what is your exact question, dear? So Ankita, your question will be all the questions related to proteins will be clear in the protein class that we will do. Uh, yeah. So what is your question with the Naveed? So this is the slide. How I use data to reach that part. Okay. I will write it down in my next lectures. I will show you my PhD work I did with the help of LCMS, how I reached to my data. So basically, it's you have to. Um, it's all done with the help of Peak software, and it's done automatically. You just have to insert your samples into your machine, and then it will automatically give you these kinds of results at the end that I have showed you. The Peak software. This one, it will show you these peptides have been identified, and then you can create a structure out of it, or you can look for the post translational modifications, you can look for ubiquitation or anything like that, or you could see some mutations that are happening in your protein. You can compare between healthy cell and cancer cells peptide mass spec data, that what is happening between them. You can check between hypoxic and in a hypoxic conditions, what is happening between the non-hypoxic and hypoxic conditions. So you can check many results, compare, and can come to the point that you are looking for. So this was just one example showing you how things are done in a straightforward manner. But when it comes to reality, it's a little bit complicated, not much. Yeah, I will share with you all slides. Don't worry. I will share with you all slides. So students, just give me 5 minutes break. Then we start with the protein part because you have a lot of questions with the protein part. I can see. So many things will be clear with that then. So don't worry. So next slide would be. So we are done with the day 3 work. So today is day 4. Protein isolations, quantifications, and purifications. So just wait for 5 to 10 minutes, then I come by and then we start again. So, protein isolation and purification, quantification. So, we'll be dealing with that part. Assay homogenizations, fractionations, centrifugations, quantitations, chromatography, electrophoresis. So, we'll be dealing with all these parts today. So, who are the Nobel Prize winners on the chromatography and electrophoresis? So, they were first Archer John Porter Martin in 1952. They discovered the partition chromatography. Same with the Richard Lawrence Millington Synch. They discovered in 1952 partition chromatography. And later, William Corin Tesselus in 1948, they discovered electrophoresis into the serum proteins. Then spectrometry and sedimentations, mainly three people, that is John B.F. Fenn, Koichi Tanaka, and the Theodor Svedberg. So all these three people, they discovered uh, spectroscopy and sedimentation. Nobel Prize winners on organelle fractionations. Albert Claude, Christian de Duveau, and George Palade. The three people, they share the Nobel Prize winner on finding out the organelle fractionations. And recently, for the CRISPR in chemistry, there are some scientists, they have found out the CRISPR Cas9, and they got Nobel Prize winner for the same also. And then in protein sequencing, Frederick Sanger from Cambridge, and from both from Cambridge. They discovered in 1980s their work, sorry, in 50s and 80s for the field of science. So, to identify an assay, this is a protein assay to identify a biochemical or biological means to use to monitor the presence of target proteins. For example, enzyme activity, specific labels, functional properties such as bindings could be identified in that case. So lactate is converted into pyruvate with the help of lactate dehydrogenase and producing NADH and H plus ions at the end. Yeah. Whereas EL5 precursor in the presence of deoxyhyposcine synthesis and deoxyhydroxylase. So they're producing both two different out products. Deoxyhypoposyl EL5A and hypoposyl EL5A2. Then how you can isolate these proteins? Mainly you can do the homogenization first step, that is disrupting your cells, and then you can do the fractionations, that is crude separation of homogenates into the fractionation. Homogenization is done with the Downs homogenizer, sonications, osmotic shock, French press, uh, and then homogeneate forms are done. Centrifugation is done at 500 G for 10 minutes. Then you check the supernatant. At the first supernatant, you will see the palette, but in the palette you will see nuclear fractionations. Then you go for further 10,000 G for 20 minutes. Then you see palette mitochondrial in the palette mitochondrial fractionations. Then you go for further 100,000 G, you see for 1 hour, you see palette microsomal fractionations like this. So, so homogenization is done with the help of Downs homogenization, sonications, osmotic shock, French press, and fractionations could be done with the help of ammonium sulfate precipitation, differential centrifugations, or by pH or by heat. Then to isolate your protein, you can also use centrifugation as a technique that we have discussed previously. Differential centrifugation techniques are present. One is with the more dense particles, the one with the less dense particles. This is a centrifugal force is given to it, to the outwards part. Then particles settle according to the mass. Yeah. Then stop the centrifuge. Then you will have the pallet, and then in the pallet is your container. You have your protein inside it. On the other side, you can do is you can add your large particles with the smaller particles on the top, and you can differentiate between large and small particles with the help of sedimentation coefficient. You can remove your samples, and you can have increasing mass of particles like this difference. Protein isolation centrifugations. Uh, So you can see that sedimentation coefficient is equal to the mass 1 minus sigma 5 divided by F is equal to UTW square X. So this is just sedimentation coefficient, which is equals to the 1s equals to the 10 raised to the minus 13 seconds. And 3000 G multiplied by is equals to, for 5 minutes, if you do for 3000 G, you will have large organelles. If you do it for 10,000 G, you will have 10 minutes. You will have palette of mitochondria inclusion bodies. At 40,000 G for 30 minutes, you will have membranes, plasma membranes, tonoplast, microsomes. At 200,000 G for 16 hours, you will have large protein complexes. Then at 400,000 G, you will have proteins of 50,000 Dalton. So this is a sedimentation velocity technique. This I have shown you in the previous lectures. You just have a low density solution, you have a high density solution. You centrifuge the tube like density gradient like this. Layering of sampling is done. Then you have a rotor. Then separation by sedimentation coefficient like this. Then you have three different fractionated, and in the whole bottom you will have your results at the end. These are the various sedimentation velocity techniques of trypsin inhibitor, cytochrome C, ribonose nucleus. There's Sedgwick things. And this is the sedimentation coefficient at the bottom, and this is the density on the left. And you can see RNA, DNA, ribosomes, various values with respect to their sedimentation velocity technique. So we did the isolation part. Now comes the quantitation part. So I will stop it here. So we will make the session of today for 9:30 only. So but mainly the sedimentation coefficient technique is equals to the 10 raised to power minus 13 seconds, and that you have to remember that one sedimentation coefficient is equal to 10 raised to power minus 3 seconds. Yeah, if you are not doing it centrifugation, then you will have the pellets might be diffused in your samples, or you will not see different chains of your samples. Yeah. So if you have any further questions, please ask. Uh, So you are not able to understand anything. Good evening, Edufabrica team. A couple of us have an issue with how the teaching is going on, especially today. So I can see that a lot of you students are not able to understand me, whatever I was teaching you. I'm sorry for that to hear this. Let's try to remain for 2 minutes. It is more like reading the presentations. Please explain the terms properly. Totally agree. The session is running too fast. Please be slow and help us to understand the topics properly. Agreed. Hia, Sagrokana, Manucharita. Okay. So students, I think problem is here basically is the topics is way too complicated and because if you have seen my previous presentations, where can I show you? Okay, I will not say anything on that issue. Um, so maybe it's you are in the first year or so that you're not able to adapt to this type of coaching because this is how we normally do it. And I think I don't know how many students of you are not able to understand my topic of today because this is proteomics is not an easy part. It's the most advanced technology that being pertaining in our field. And I'm really sorry if I have been to given my best way to teach you, and you're not able to understand each part. Uh, so I can't. Yeah. Because, Pacha, the thing is, these topics that you are, because initially when I start with today was the basics of protein stability. Then we continue with the further part of mass spectrometry, which is quite advanced actually for all of you. And then I went for third part that is protein quantification. And this I think you will not able to understand at all if I because you all doesn't know anything about proteomics so far. I have a feeling, and you have taken the wrong course because most of the other students they are with me and they are continuing with us. Maybe you can exchange your. Are you part of MBBT also? So maybe you should think of switching from bioinformatics to MBBT. So that is more easy to go for, and go for that because proteomics. Yes, sorry, I can't. What about others? Yeah, yeah. Okay, okay. Hmm. Okay, I will take your point in. That's a good suggestion. I can understand that. So thank you, Nagesh. Thank you, Hia. So I can, what can I see is now that you all want to understand the basics of proteomics, not the advanced versions. So I will try to make more lectures more easy and more understandable, and you can understand more principles. So maybe give me, yeah. So at the moment, we will continue with these lectures of tomorrow onwards about protein isolation and purification. And so we continue with the same. It's a lot of interactive session actually, and it's a lot of virtual lab also inside that, so that you are aware of how protein isolations and everything uh, is done in that way. So problem is that you're not updated with the basics of proteomics. So and try to be like, let's work as a team, student. So I would be happy, like you gave me these suggestions today, so that I can start working on them for you, because these lectures are meant for you only, not for me. Because I have the knowledge, and my reason as a teacher is here to teach you uh, whatever the knowledge that I have, and I can share with you. That's the main purpose of a teacher. And you as a student is here to grab knowledge as much as possible from us. So the terms that I'm using here, I'm using, I think is quite way beyond that your level here. Okay. So no worries. Let's start fresh start tomorrow also with the new topics of isolations. And I can share with you also that today's recording. I think my recording is going on. I think because I'm not trusted with my YouTube recording so far. But if it is going fine, then I will share with you also this one. So no worries. I will try to maintain it more easy for you. Okay, student. Then we end the session here. Yeah, yeah. We will end the session here. See you tomorrow. Uh, give you some materials about more basics part with the proteomics only. So don't worry. Just chill around. We see us tomorrow then. Good night. Sleep well, and I will work for you. So you got queries. If you want to quantify total protein, then centrifugation is done at which RPM? Want to quantify total protein centrifugation at बच्चे, I can't tell the RPM at the moment, but it depends. RPM depends upon your size of a protein. Whatever the size of a protein is, it's directly proportional to your RPM. So according to that, you can do it. If you want confirmation about one particular protein isolated, then how to purify it, by which method? So you can do it with the STS gel, with 1D gel. You can run one STS gel on a STS page, then with the help of molecular weight, you can remove that gel part and trypsinize it and go for mass spectrometry, LCMSMS. Nano is 10 raised to the minus 9, but the liquid chromatography level of LCMS would be done at the very nano level in that case. Okay, then students, we see us tomorrow with the basics, and I have my recordings with me also. I try because I'm not sure my recordings will be done, but it was done, so I'm happy. So I will share in the group. So we try to improvise your syllabus now because you are not able to understand what we are gonna teach you. So it will take some time to absorb this and to start something new for you, all of you. So, okay, then see you tomorrow. Good night.